What is color blindness? How can we see the color spectrum and what happens when people can't see in full color?
In order to understand the neurophysiology behind the color blindness, first we must understand the eye and how it works. The eye sits in the socket named the orbit, which is attached to six extraocular muscles. The actual eye is made up of the cornea, where the light is focused through and bent and refracts. Behind the cornea, there is fluid space called the anterior chamber, which is filled with aqueous humor. They maintain a constant eye pressure. Behind the chamber is the colored part of the eye, the iris which surrounds the pupil, with muscles responding to stimuli widen or constrict the iris to control the amount of light entering the eye. Behind the pupil is the lens. This focuses the light to the back of the eye, changing shape to focus on the object up close. The focus light is directed to the retina, that has the light sensitive tissue to gain information about what is being seen. A small part of the retina, the macula, is responsible for providing detailed central vision. The other part is for visualized periphery. The retina sends light as electrical impulses through the optic nerve to the brain. The optic nerve is made of millions of nerve fibers that transmit these impulses to the visual cortex, the part of the brain responsible for the sight.
Each retina has cells called photoreceptors. They are responsible for changing light into energy signals. There are two types of photoreceptors, rods and cones. Rods perceive black and white, enabling night vision and respond to motion, while cones are responsible for seeing different wavelengths of color and respond to light. The three main color receptors are blue, which respond to short wavelength, green, which responds to mid wavelength, and red, which responds to long wavelength. Each retina contains 6 million cones, which deteriorate with age. From a neurophysiological perspective, photoreceptor cells have specific proteins that allow for the light to be properly converted to biochemical signals. The protein is known as rhodopsin, is responsible for the transformation.
The phototransduction process is the biochemical process of the photoreceptor cells in the retina that convert light into electrical signals. When photons of light strike photopigments, example the rhodopsin in rods, this triggers a conformational change that activates the G protein called transducin. The activation of transducin leads to a cascade of reactions. This causes sodium channels to close in the photoreceptor cell membrane. Closing the sodium channels results in a hyperpolarization of the channel, and the inside of the channel becomes more negative. This causes the decreasing of glutamate, which is a neurotransmitter that regulates the activity of bipolar cells. The bipolar cells are responsible for transmitting the signals to the ganglion cells. The ganglion cells then send visual information via the optic nerve to the brain for information processing, and therefore the brain allows us to perceive images.
Each retina has cells.
Color blindness, also known as color vision deficiency, is a relatively common condition where individuals perceive colors differently than the typical way. This occurs when the cones, specialized nerve cells in the retina of the eye, fail to function properly. These cones are responsible for processing light and images as they enter the eye and sending signals to the brain, enabling the perception of color. However, color blindness doesn't mean a complete inability to see colors, as the majority of people with this condition can still perceive a range of colors, but they may perceive certain colors differently or struggle to distinguish between specific colors and shades. However, in very rare instances, some individuals may be unable to see any colors at all. For most people, color blindness is an inherited condition, passed down through the genetic factors from biological parents. The most common forms, such as red-green color blindness, are typically inherited from the mother. However, color vision deficiency can also develop later in life due to medical conditions, medications, or other external factors. Cones are nerve cells in your eyes that can detect colors in the visible spectrum of light. This spectrum includes all the wavelengths that humans can see. These range in length from 380 nanometer cones, short cones, or to 700 nanometer cones, long cones.
Anomalous trichromacy is a type of color vision deficiency where all three cone types are present, but one is less sensitive to its specific wavelength range. This reduces color differentiation with mild cases affecting pale colors and severe cases affecting even vivid colors. The X-linked gene SQ28 codes for opposite in L and M cones, which normally respond to specific wavelengths. In anomalous trichromacy, genetic defects made these cones respond to the broader range, leading to color confusion, such as red sensing cones reacting to green frequencies. Dichromacy is a color vision deficiency where one type of cone is completely absent, leaving only two functional cone types, X with either L or M. This limits color perception to the wavelengths detected by the remaining cones, making it difficult to distinguish fully saturated colors. The absence of a cone is caused by genetic defect in the X chromosome. Monochromacy is a condition where a person has only one type of cones or lacks cone function entirely. This results in little to no color perception, causing the world appear to shades of gray.